So thanks, Erica, for the really nice introduction and a great overview. And particularly want to thank the organizers for giving me the opportunity to share some of the work we have been doing that was deeply motivated by Clyde's insights and the concepts that he developed. It's a real personal honor to be asked to present this here at the homage to Clyde Herzmann. Couldn't ask for a better venue to really share with you what I want to talk about, which is basically how epigenetics might serve to embed some of the early life experiences that we heard earlier about today. So, how do I do this? So, I'm sure all of you know that our life is a continuous development, but what really fascinates me and many others, and including Clyde, is that these early years of life are really years of wonder in many different ways. But one particular way that I think is extremely fascinating is brain development. So if you look at this slide here, what you can see, these are sort of neurons going from newborn to 1 month, 9 months, 2 years into the adult, and you can see that there's a wondrous, fascinating dynamic in that these neurons are created. If you look very careful, you can see that the acceleration in this creation between 1 month and 2 years is just fascinating. But if you look careful, you can actually see that there's then some pruning going on because the adult actually has much less neurons than a 2-year-old. Whether that means that a 2-year-old is smart or not, I put it out as one of the provocative questions, I guess. But really, brain development in children is an absolutely fascinating area. And you can see here some of the data so that, for example, in the first years of life, the brain grows at a pace of more than 700 new of these connections per second. Okay? We'll never ever get this in later on in life. And in fact, if you're my age, they probably die off. By 3 years of age, a child's brain is twice as active as an adult brain. I have actually a 3-year-old son, so I can attest to that. Thinks much more quicker than I do. But most importantly, really, in the context of Clyde's legacy, is that these early life experiences that determine the lifelong capacity of the brain. And you've seen many of these kind of slides, but one that I think is still very striking is this stratification of vocabulary in children that are 3 years old that are born to parents on different levels or different levels of the socio-economic status, where you can see children that are born to parents on welfare have lower vocabulary than children that are born to middle-class, working-class families or than professionals. So there's really a dramatic stratification that happens in this particular case on the ability to learn, on the ability to use vocabulary that is dependent on the childhood environment, in this particular case, socioeconomic status. And what we have been really focusing on and inspired by many of the, as I said, concepts that Clyde has put out, how actually does that happen? What are the biological underpinnings of these social experiences affecting our brain development and potentially our health across the lifespan? And the field that we're working on is called epigenetics. So does anybody speak Greek in here? So epi actually stands for above genetics. And so we always look smugly down on the geneticists. But what epigenetics means, you can see it here, it basically refers to the persistent and potentially heritable alteration in the genomic information that do not involve changes in the DNA sequence. So it's not the kind of gene sequence that you're given by your dad or your mom at conception, it's something else. And the particular area that we really are most passionate about is called social epigenetics, which really then refers to the process by which these life experiences, such as the early life socioeconomic status and others, influence the ways by which our genes are expressed. And what the paradigm that we then like to look at is how do experiences affect biological pathways and affect gene expression. So we actually like to say we go from society to cell as opposed to cell to society, contrast to what most people do. But of course, we are keenly aware that this in turn then these changes in gene expression might feed back into biological pathways that affect our experiences. And so key to understanding all of this is then this word here, gene expression. I know this might elicit some nervousness in the audience, so I I want to make sure that you follow me with, it's actually really simple. What I like to think about as a gene is literally a light bulb, okay? And so we have, as you probably know, well, as you can see here, you have light bulbs that are totally off or light bulbs that are totally on. If you live in a fancy house and you've got a dimmer, you have light bulbs that are sort of anywhere in between. And so think of your genes. You have 25,000 genes, a copy from the dad, a copy from the mom. So what I really want you to think about of your genes as of 25,000 light bulbs, okay? The epigenetics then actually is the dimmer, okay? So because what the epigenetics does, it sets the activity of these light bulbs. And as you can see here, the dimmer is all the way up and all the way down, respectively, and that sets the activity of the light bulbs. So the epigenome literally are dimmer switches, simple as that, and the genes are the light bulbs. So how does that work? So for example, let's say we have a gene that is totally turned on, a light bulb that's totally turned on. This is the DNA. Now what actually regulates this dimmer switch? It's a number of different things, but the one that's best studied, and I'm going to focus on today, is a small chemical group called a methyl group. You probably have some fuzzy memories of high school chemistry. A methyl group is basically a very simple group of a carbon and 3 hydrogens. And what can happen is that these methyl groups can be attached to the DNA itself. And watch what happens when we attach a methyl group to the DNA. You see, the methyl group comes in, the dimmer goes down. We get more methyl groups, dimmer goes further down, and we got another methyl group, and the dimmer is all the way down, and with that, the light bulb is turned off. Okay, so that's as simple as it becomes in epigenetics. That's really what it's all about. And so to summarize this, we can have the light bulb fully on, dimmer up. This is called an active gene. Or we can have the other extreme, a silence gene, where we have the dimmer all the way down and the light bulb is off. So that's the sort of circuitry between the epigenome and the gene activity that is really crucial to understanding the epigenetic biology. Now I want to give you what I think is still perhaps the most instructive example of epigenetics at work in the next slide. And this is shown here. So if you look at this, you see 5, these are actually mice. Is there anybody in the room who thinks these mice look alike? Good, because otherwise we'd have you send you to an optometrist. So obviously there are these yellow mice and these brown mice over here. If you were to see this kind of picture, what would you think? Like, what's the cause for these mice, one of them being yellow, the other one being brown? It's your genes, right? You're born, like my wife is blonde, my son is blonde, I'm brown-haired, so he got the genes for blonde hair. Well, what I'm going to tell you that these mice are actually genetically absolutely identical. So they have been bred by these mouse geneticists to be a clone of each other. They're absolutely identical, yet they look very different. So the question becomes, why do they look different? Well, let's look at the next slide. The reason that they look different is that there is a dimmer switch and light circuitry that's different in each of them. You can see the yellow mouse, the dimmer is all the way up, the light bulb is all the way on. And as we go over to the brown mouse, the dimmer is pushed down, the light bulb is turned off, and the mouse is brown. Okay, so it's this circuitry between dimmer switches and light bulbs that underlies this code color in these mice. Now, the really fascinating thing about this, what sets that dimmer switch in the first place, and it turns out it's actually the nutrition of the mother during pregnancy. And there are a number of different nutrients that have been shown to affect this dimmer switch setting. But to me, this is a beautiful example of epigenetics, because what happens is that somehow the experience, quote unquote, during pregnancy, the nutrition that this fetus, the growing fetus, was exposed to, got literally under the skin to affect the dimmer switch setting and to affect the light bulb, and then to ultimately change the coat color in these mice after they were born. Now, of course, you might argue, coat color in mice, really not all that exciting. Fascinating, but what does it do for us? Well, watch what happens if you actually let these mice grow up. Okay, now let's just focus on the yellow and the brown mouse. So this is what the next picture that I'm going to show you is the same mice at the equivalent of 30 to 40 human years. Look at this. Brown mouse, lean and fit. Yellow mouse, it's huge, it's obese, twice the weight of the brown mouse, and it develops all kinds of health-related problems, metabolic disease, depression, you name it. And this can be solely traced back to the nutrition of the mother during pregnancy. So to me, most beautiful example of epigenetics, you get an environmental cue, that being the maternal nutrition, that sets the dimmer switch, affects the light bulb setting persistently all the way into adulthood to profoundly affect health and behavior across the lifespan. Is this actually something that's of relevant for human biology, for human population health? Well, it is, and there's a numerous study that show that. The most famous ones, of course, as you probably know, talking about the Dutch famine at the end of World War II, where women grew up in 500 kilocalories a day. There are a couple of very, very interesting papers that suggest that in people that were born, so 60-year-old people that were born during the Dutch famine, there is a difference in their dimmer switch setting in their white blood cells that is related to growth, to genes involved in growth and also in behavior. There's also a number of studies that suggest that prenatal smoking, maternal smoking, can affect these dimmer switches in the children in human populations. Seminal work that originated here at McGill in Montreal with Michael Meany and others, suggesting that the quality of the maternal care affects the epigenome, the dimmer switch setting in the offspring. I'm going to touch on this from our own work that we did with Glide in a little bit. And lastly, the closest, of course, we can come in human population to these kind of clonal identical mice are identical twins. Many of you probably know identical twins, so even though they have exactly the same genome, more or less, they're often developing health-related problems at a different time or a different severity. And interestingly enough, it has been shown that the epigenome of identical twins, the dimmer switch setting of identical twins, is almost identical in young identical twins, but it diverges in twins that are older, and in particular if they had been separated early in life. So collectively, this then leads us to an integrative model of epigenetics in that we believe that epigenetics acts together with the genome, but also with the environment to really, hopefully, in some way or another, affect healthy starts and in some ways allow us to prevent disease in the long run. And we really have been motivated a lot by this kind of insights from the animal models and the limited human studies to ask the question, how do these early life experiences mechanistically get under the skin, and what can we learn from this? And so we have been focusing on things like socio-economic status in early life parental stress and their effects on the epigenome and how that ultimately might affect childhood temperament behavior and trajectories. And one of the beauties of being an epigeneticist is that you get to work with a number of fascinating, wonderful people. And I really have to say that I've been standing on the shoulder of giants to do this kind of work. So we have been working with Craig Miller, Edith Chen, and Tom Boyce, all of whom were at UBC but moved south to either Chicago or San Francisco, and Marilyn Essex at the University of Wisconsin. But most importantly, I really want to dedicate these next few slides to Glide Hertzman's legacy, because Glide has been a personal mentor, a role model, and a friend, and really has been the person in many, many different ways that has allowed us to do all this work and has been fundamental in us developing these research questions and these approaches. So I really want to dedicate this to his legacy. So as I said, collectively, our research program has addressed some of these issues, and I want to show you some data. Now, just to go back again, and I think I'm preaching to the choir here, of course, but there is numerous data that suggest that, for example, early life socioeconomic status affects health across the lifespan. One study that I in particular like is the Johns Hopkins physician study, where more than 1,000 white male physicians were followed after graduation, and as you probably can appreciate, they graduate from Johns Hopkins, they're very wealthy, they're well respected in the community, but nevertheless, at the age of 55, there is a stratification in their comorbidity, in their morbidity to coronary heart disease, in that physicians that grew up in low early life socioeconomic status had a 2.5-fold increased risk of having severe heart disease. And as a geneticist, or somebody with genetic training, this is actually in the ballpark of effect size that's very similar to what you would expect for some of the very severe genetic mutations. So then this really raises the question, how do these experiences get under skin? And Glide, this might not be so well known here, Glide really has written some fundamental pieces that has shaped the field, one of which is this review, I think, in the annual reviews that talked about the biological embedding of early life experiences and its effects in adulthood. And being the sort of visionary that he was, he postulated at a time that it might be actually epigenetic mechanism, so this circuitry between dimmer switch and light bulb that might be at the root of how these experiences get under the skin. And so we tested this directly in a number of studies. The first one where we had a cohort of adults that was basically either from low or high childhood SES and mixed for their current adulthood SES, thus allowing us to directly derive any findings about childhood SES that are not confounded by adulthood SES. And what we did in these studies, we measured basically the dimmer switches and we measured the light bulbs. And so without going into too much technical detail, we found that there are indeed light bulb differences between the two groups. And there were just random differences. There were differences that suggested that genes that are involved in inflammation had different light bulb intensities in the people that grew up in low SES compared to the people of high SES. Consistent with that, if we then take immune cells from these people and challenge them with various agents we can find here, this is shown over here in these two graphs, we can show, we find an enhanced immune response in the people that grew up in early life low SES. And furthermore, we also find increased cortisol output over the course of the day in people that grew up in low early life SES. And lastly, as shown by these skewed p-value distributions over here, we find a correlation between these dimmer switches in these white blood cells and the early life SOS in these people. Interestingly enough, and I pose that as a sort of provocative question, we do not find any correlation with current SES. So the dimmer switches that we carry in us are much more related to the way we grew up than, at least in socioeconomic status, than the socioeconomic status that we have now. Collectively, this data suggests that indeed there is a biological residue at the level of the dimmer switches, at the level of the light bulbs that's functionally expressed in heightened immune response and physiologically in heightened cortisol output during the day that is caused by early life socioeconomic status. Of course, to some extent, that is somewhat of a depressing message. We I always tell this story. I got a call from a Vancouver newspaper when we published this paper, and I was fairly new to this, and I sort of have a nice chart with the reporter. And the next day, this is a newspaper that's read by it's not the most high, everyday people. And I was quoted as if their readers were doomed, which is what I didn't say. But out of every challenge comes an opportunity, and the opportunity that we took is like, is there anything in this group of people? Is there anything in their light switches, in their dimmer settings, in their light bulbs that could provide hope, could provide opportunities for intervention? And I wouldn't be here if that weren't the case. So what we found actually is that in people of low early life SES, there was actually resilience to these effects if they reported and if their parents, this was done by self-report, but also by interviews with the parents, if there was a high level of maternal warmth. So this can be seen on these light bulbs. We can see that on the dimmer switches, but perhaps most importantly, we can see it here on the reactivity of the immune cells to challenge, where clearly with the people that have low maternal warmth, experience low maternal warmth to high maternal warmth, as a difference in many of these readouts that we look at in terms of their immune system. Okay, so basically, if you happen to grow up in this kind of circumstance, but your mom loves you a lot, you're going to be okay, at least on the level of this epigenetic embedding. So, and another one of my provocative statements, I always advise people to choose their parents wisely. And so, this study was published 2 years ago in Molecular Psychiatry. Part of the problem is that, of course, we're retrospectively ascertaining maternal warmth and childhood environments, even though the parents were interviewed. Being a new parent myself, I don't think any of us would ever admit that we're negligent parents or anything like that. And so, again, out of a challenge comes an opportunity, and the opportunity was to facilitate it through GLIDE to work with Marilyn Essex at the University of Wisconsin. And Marilyn has made a living, as some of you might know, on a longitudinal birth cohort called the Wisconsin Study of Families and Work. And what Marilyn had collected at the age of 16, were DNA from children in her cohort. And so we used this opportunity to basically ascertain their dimmer switch setting in these children and ask, how is that related to their early life environment? Of course, we're interested in socioeconomic status at first, but unfortunately for us, or fortunately for those children, this was a suburban Wisconsin cohort. There was no socioeconomic status gradient that we could really make use of. But there was quite a gradient in terms of the level of stress that the parents, that there was in these families. And what we found in this study, this was published in Child Development, I think 2 years ago, or last year, that there was actually, there were interesting correlations. So the first one was that we found in this adolescence a correlation with maternal stress during infancy. And that was true when we kept both girls and boys together. These are 109 kids. We lost some statistical power when we separated them by boys versus girls. We did not find really strong signatures for paternal stress at this developmental stage. In contrast, when we looked at preschool, now we didn't really see anything with the maternal stress, but we saw a very strong signature of paternal stress, somewhat in the entire group, but much more pronounced by these numbers in the girls only. And the HCS versus MCS are basically just ascertainments of statistical significance in these based on multiple testing corrections. And so really what this shows is that indeed in this adolescence we can see a vestige of parental stress that is specific for the developmental stage, but it is also specific for whether it's the mom or the dad stress. Now I guess another question I was actually asked, and I want to put out here at a recent talk in Munich was, is this perhaps just really a reflection that back in the days it was the moms who stayed at home during infancy? What would happen and if you were to look in families where we have a stay-at-home dad, would it now be the dad? So is it really just matter who is at home as opposed to it being the mom? Is that just a reflection of public policy? So that's something that I think is very interested in. But just to summarize then, I hope that in these 20 minutes that I think I maintained, I showed you that really gave you just a flavor for why we think epigenetics is such an integral component of human health, disease and behavior, and shared with you some of the work that was deeply motivated by Clyde's insight, by his mentorship and by his leadership, that talks about the genomic embedding of early life experiences, how epigenetics, these dimmer switches and the light bulb, the circuitry, really is a biological residue of early life poverty, how it can be associated with early life parental stress, and I didn't have time to show you, but in a current study that we're just finishing, we're finding that perhaps not surprisingly that childhood temperament is also correlated with the setting of these dimmer switches. And so that really then leads me to say that perhaps the biology of early life experiences and the mechanism of epigenetic embedding might provide us opportunities to provide basically the best healthy starts to our children to prevent disease and, somewhat in a challenging statement again, design perhaps targeted interventions. And just to acknowledge all the people who've done the work, I think I mentioned them, I do want to highlight that this has been becoming a fairly large research program, and it was really enabled by a fairly small grant, but nevertheless that Clyde gave us 5 years ago, because he had the foresight that this would become an important area of research. And I want to also, of course, acknowledge NIH, CIHR, and the Canadian Institute for Advanced Research and the Mumafakin Foundation who have supported all of this. And so just want to leave you perhaps with this question, is it nature versus nurture, this old paradigm that everybody talks about, or both? Thanks a lot.
Okay, that's great. Thank you.